so much. I hope you all just join me in taking a moment to thank all of our wonderful presenters and poster presenters from this poster session. Like, everybody has done such a wonderful job today. So thank you. Yes, I am Erica Hutchins. I am an assistant professor in the Department of Cell and Tissue Biology, and I will be chairing the session today. I'd like to present our first speaker, Tess Foote, who will be speaking on the impact of preventative oral health policy on country-level dental health status.
Hi, everyone. So to provide a little background to my research, you should know that according to the WHO, dental caries in permanent teeth is one of the most common health conditions, despite being preventable and treatable at early stages. This is partly because dental services have often been excluded from the global movement towards universal health coverage in many countries. And this is all very surprising given that there's ample evidence that supports the positive effects of preventive dental services on oral health. Preventive dental services have been shown to reduce decayed, missing, and filled teeth indexes in children and have also shown to be cost effective and cost saving. Studies show that patients who seek treatment for preventive care have lower dental costs than patients who seek treatment for problems alone. Although there's ample positive evidence on the effects of preventive dental measures, there's still a low utilization rate of these services in many countries, and this is partly due to inadequate regulation of these services and inadequate insurance coverage. So the overall goal of my study is to assess the association of regulation of preventive dental services with oral health outcomes and oral health expenditures on a global scale, and we'll be looking at member countries of the Organization for Economic Cooperation and Development. So how did we do this? After accounting for missing data, and there was a lot of missing data, we included 19 of the 38 OECD countries, and here's a list of the countries included. So the variables we're looking at are oral health outcomes, oral health expenditure, and oral health policy. Oral health outcomes were measured using DMFT indexes from the WHO Oral Health Country Area Profile Project database. And the parameters set were children aged 12 to 18 years, because this is the age group that most preventive policies are geared towards. For oral health expenditure, we measured this as a percentage of each country's gross domestic product, and our data source was the OECD Health Expenditure and Financing database. And for both of these variables, outcomes and expenditure, we calculated a mean from the years 2010 to 2019 to have an average from that decade. And for the oral health policy research, we did country-specific research for each country. And after reading all of the relevant policies and guidelines, we came up with these 3 binary variables as prominent indicators for regulation of preventive care. So the first variable is available dental services for children. And this claims that the preventive services are included in a country's national insurance scheme or public dental service. And then mandatory dental services for children. And this shows if the country has a specific law that actually mandates that children receive this care. And then guidelines and technical standards shows if the country publishes clinical practice guidelines for their dentists on how to actually provide this preventive care. So our results. As you can see, our most common policy was available dental services for children, with 78.95% of the countries having these available services. And our least common policy was mandatory services for children, with only 26.32% of countries having an actual law that mandates children receive this care. And this indicates that in many of these countries, resources are there, but they're not necessarily being mandated, and this could contribute to why they're often underutilized. This is a correlation matrix showing the relationship between each of our variables, and bolded are the relationships that were found to be significant. So there's a relationship between average oral health expenditure and average DMFT index, and that's to be expected. As spending increases, oral health outcomes also increase. And our oral health policy, the only policy which had significant relationship with average DMFT index and oral health expenditure, was the category of mandatory dental services for children. And what these numbers suggest is that with mandatory dental services for children, there could be an association of a 1.32 reduction in DMFT index and a 0.16% increase in oral health expenditure. And then this graph is just further highlighting those relationships, showing average DMFT index versus average oral health expenditure. And the countries in blue, Denmark, Finland, Germany, Japan, and Sweden, are all the countries that mandate dental care for children. And these countries are seen to have lower DMFT indexes and higher oral health expenditures. And initially, this was a little disappointing to find because I was hoping that with more regulation of preventive dental care, that oral health expenditure would decrease. But that's okay because these countries have also been said to have very efficient oral health systems in a study that weighed a cost benefit analysis, suggesting that even though they have higher oral health expenditures, the slightly higher spending may be worth this significant reduction in DMFT index. And these graphs are just further illustrating those relationships. In figure 2, you can see countries with the policy have significantly lower DMFT indexes. And in figure 3, you see that countries with the policy have higher oral health expenditures. I'd like to talk a bit about some country standouts. So Spain and Switzerland, these were countries without any policy in any of these categories. So I was looking for trends between the countries, and both of these countries operate under highly privatized oral healthcare models. Spain specifically, the services provided vary drastically by region. So it was kind of hard to look at a national oral health policy because, as I said, it varies based on the region. And in Switzerland, dental coverage is not regulated differently for vulnerable groups like children. So oral health policy pretty much looks the same for the general population, regardless of age or disability. And then on the other end of the spectrum, Denmark and Japan, these were countries that had policies in all of the categories. And as mentioned before, they ranked highly on a study measuring the efficiency indexes of countries' oral health systems. And in both of these countries, dental care should be free of charge for children. Another similarity was that both of these countries have their oral health care systems tied very closely to their schooling systems. And this was a trend I saw in all of the countries that mandate dental care for children, suggesting that this could be an outlet or like the first contact point for children in the oral health care system. So to conclude, mandating dental care for children is associated with a 1.32 reduction in mean DMFT score and a 0.16% increase in oral health expenditure. My general takeaway from this study is that preventive resources are often available, but underutilized and inaccessible. And policy implementation can change this. Policy can be implemented to increase the regulation of preventive services and to ensure that this care is being reached to a wider population of people. I'd like to thank the UCSF School of Dentistry Global Oral Health Program for funding this project and my mentor, Dr. Tracy Lynn, for her tireless mentorship throughout the duration. Thank you. Here are my references.
We have a question, and there's a microphone.
What's up, Jess? Thank you for that great presentation. My question for you is, in your study, why did you only focus on OECD countries?
That's a good question. We focus on OECD countries because this is a group of countries that are mostly democratic and higher income countries, and they work with governments and policymakers to establish more evidence-based policy standards. So with that in mind, I figured it would be easier to conduct this research with countries that have more standardized policy. Throughout the projects, I realized that even in this group of countries, that was like not the case, which really highlights the need for more standardized oral health policy globally for more studies like this.
Thank you. Just one more question. Thank you.
Phenomenal presentation, Tess. I want to ask, so what are the next steps for your project?
So this project only focuses on the existence of the policy. In the future, I'd hope to look more at the specifics of the policy as well as how the policy is actually enforced within the country. So yeah, that's my hopes for the future. Thank you.
Great, thank you so much. Thank you, Tess. I'd like to now present our next speaker, Leah Sedgi, who will be speaking on determining the effect of processed grain or high fiber diets on microbial and immunological markers of periodontitis.
Periodontal disease is a chronic inflammatory disease that is characterized by soft and hard tissue destruction of the periodontal complex. The initial stages of the disease, or gingivitis, are largely reversible if treated. However, this disease state often goes unnoticed and evolves into periodontitis, which is an irreversible form of the disease, and this can lead to tooth loss or dentulism. This is problematic because nearly 50% of the US population over 30 years old suffers from some form of periodontitis, ranging from mild to severe. And of these individuals, 65% are below the poverty line. As I mentioned, periodontal disease is irreversible, and periodontal treatment is very expensive and inaccessible to the majority of those that are afflicted with the disease. As such, it's important to discover new means of periodontal disease prevention that are widely affordable, accessible, and impactful on a large scale. To do this, it's important to understand what are the drivers of periodontitis. So periodontal disease is primarily driven by an interplay between the oral microbiome and inflammation. The oral microbiome builds sequentially via metabolic and physical interactions amongst the microbes, which eventually leads to the integration of periodontal pathogens into the system. And concomitant with this, we have an increase in inflammation in the periodontal complex. So ultimately, dysbiosis and inflammation interact to lead to the disease state. So how can we mediate these drivers of periodontitis in such a way that's largely accessible to prevent the disease? So my approach to this is to study populations, whether past or present, that have a low incidence of periodontal disease, albeit not having access to modern oral hygiene practice or dental care providers. And an especially interesting time point to me is the Mesolithic to Neolithic transition at the advent of agriculture, at which time point grains were introduced as a main staple of our diet. And with this, we also see increased evidence for dental and periodontal diseases. Another important time point is the Industrial Revolution, in which processing techniques introduced ultra-processed cereal grains as a main staple of our diet. Today, modern hunter-gatherer societies, such as the Hadza people in Tanzania, display exceptional oral health status, albeit not having access to a dental care provider. And this is attributed to consumption of fibrous vegetables. Several studies have looked at the correlation between periodontal disease and processed grain consumption. However, these studies are largely correlative in nature and have not looked at the mechanistic underpinnings of this. So I hypothesized that low fiber processed grain diets drive periodontal disease by increasing inflammation in periodontal tissues and by promoting microbial dysbiosis. To do this, I implemented a polymicrobial infection model of periodontitis in mice. So I first treated the mice with antibiotics to establish a baseline microbiome amongst all of the subjects. I then assigned mice to various diet types, including whole grain, processed grain, fiber bar, and placebo bar for 4 weeks in the absence of infection to establish an oral microbiota that was reflective of each diet type. I then infected the infection group mice with a polymicrobial inoculum containing periodontal pathogens for 4 consecutive days each week for 4 weeks. And then I collected hemimaxilla and mandibles for morphological, immunological, and microbiome analyses. Alveolar bone loss is a classical clinical parameter in periodontitis, in which the hard tissue destruction of the alveolar bone increases the distance between the cemento-enamel junction and the alveolar bone crest. To measure this, we performed micro-CT and measured 4 distinct sites on the mouse mandible. And for the sake of time, I'm only going to be presenting those results that were of special interest to us. So I'm focusing here on the processed grain versus whole grain groups, but we saw that the processed grain group had significantly greater bone loss to the whole grain group in the absence of infection. And this was really interesting because it suggests that even in the absence of infection, processed grain diets are able to promote a periodontitis-like phenotype. Moving on, we next sought to understand how diets are affecting the inflammatory profile of the tissues. And to do this, we looked at the cytokine levels of various cytokines that are recognized for their role in soft and hard tissue destruction and periodontitis. We saw that between the whole grain and processed grain diets, there was a significant difference in the cytokine profiles and in the absence of infection, but not in the presence of infection. And looking more closely into this to determine what's really driving these differences, we saw that the processed grain diet had a significantly higher level of IL-17A compared to the whole grain diet. And this was really exciting to me because IL-17A is really recognized for its role in promoting alveolar bone loss. Finally, we sought to understand how diet prompts changes in the oral microbiome with relevance to periodontitis. We saw that between the whole grain and processed grain diets, there were distinct microbial profiles in the absence of infection, but not in the presence of infection. And looking more closely into the genera that are driving these differences, we saw that the whole grain diet was characterized primarily by lactobacilli, whereas the processed grain diet was driven primarily by Enterococcus species. And interestingly, in the presence of infection, the whole grain diet seemed to somewhat resemble the processed grain diet in the absence of infection. And with the processed grain with infection, we saw an increased abundance of Pseudomonas species. And while Pseudomonas are not putative periodontal pathogens, they have been noted in chronic periodontal lesions, as well as are well-recognized pathogens in other disease states. With the fiber and placebo groups, we saw that these diets prompted specific distinct microbial profiles in the absence of infection, but not in the presence of infection. And looking more closely at this, we see that these differences are driven primarily by the relative abundance of Enterococcus, which are higher in the fiber groups compared to the placebo groups. So overall, we see that processed grain diets promote increased alveolar bone loss and IL-17A compared to whole grain diets. We also saw that the oral microbiome differed significantly between whole grain and processed grain diets, as well as fiber and placebo diets in the absence of infection. Our further investigation will focus on the potential local effects, such as food texture and starch bioavailability, which are major components of my project that I wasn't able to touch on today, or distal effects of diet on periodontal disease, such as the gut microbiome. And we were able to collect stool throughout the experiment. So we will be looking at this also. And with that, I would like to acknowledge my very interdisciplinary team who has helped to make this possible. So the Capilla Lab with their focus on periodontal disease, the Lynch Lab with its focus on microbiome science, as well as Sunita Ho's lab, which is focused on biomechanics. Again, wasn't able to really touch on this, but a huge aspect of the project. As well as my funding sources, the NIDCR, as well as UCSF Center for Benioff Microbiome Medicine, and our collaborators at Better Biotics. Thank you.
Very good. Any questions in the audience?
Nice talk. I'm curious on how did you measure the cytokine levels? And then did you see a difference in the rank ligand? Is it a function of that IL-17 is increasing osteoclastic activity, or there's just more rank ligand inducing osteoclastogenesis?
So we didn't look at rank specifically. We used an ELISA-based assay that was targeted for specific cytokines. So that we chose those cytokines specifically. They weren't anything that popped up.
One more question.
Great talk, Leah. I was wondering if there are any differences in the mechanical forces that are loaded onto the alveolar bone or the oral microbiome, depending on the diet that they are given.
Yes, great question. I have measured dietary stiffness in the whole grain versus processed grain diets. And we saw that the whole grain diet had a higher stiffness than the processed grain diet. And so the differences that we're seeing really might be attributed to texture, because the placebo and fiber bar groups, they didn't really have a difference in the texture. So these differences might be driven by that.
Thank you so much.
Thank you.
Okay, thank you. Our next speaker is Nathan Young, who will be speaking on developmental morphospaces reveal novel allometric shifts in the embryonic brain and face.
Thank you, Erica. As you might notice, I'm not Susanna Bavruzovka. I am actually a faculty interloper. Suska is sick today, so unfortunately she could not speak, but I'm speaking on her behalf today. But because I'm interloping, I kind of really feel this meme today. Yeah, but I hope to match the excellence of all the speakers, of all the young, bright scholars that have been presenting their work today. So let's get into it. All right, so human development is exceedingly complex. It's mind-bogglingly complex even, in which all of us have gone through the process of one cell into tens of trillions of cells, all while maintaining functional integrity. It's incredible if you think about it. It's much different than, say, an engineered product. Importantly, because of this, we are a product of historically contingent, or we're a historically contingent process, right? So each individual event is going to lead to another event, and earlier events then affect these later events. This is no less true for craniofacial development. So we can think about craniofacial development as a series of developmental processes stemming from, say, neural crest induction all the way to skeletal growth. And these overlap in terms of their effects, in terms of time and space, and they contribute in ways both small and large to our ultimate phenotypic outcomes, which we like to think is ourselves, right? In my lab, we model complexity of development as this kind of subway diagram, thinking about it as a series of decision points, our branching outcomes. And we use a concept, which we like to call a developmental morphospace, to quantify how genetic variation or perturbations in genetic variation actually lead to phenotypic outcomes, and how these phenotypes occupy this kind of morphospace, and how earlier events are then going to lead to later events and pattern later events as well. And we're particularly focused on decision points associated with brain growth, as well as facial prominence outgrowth. So in 2014, which is granted a long time ago, we used this approach to understand or characterize craniofacial development in a large number of amniotes. And amniotes are egg-bearing animals, things like mice that we love, birds, gators, turtles, even humans are amniotes. And we looked at all these different groups and we characterized them in terms of this developmental morphospace. And what we found is that we could, when we looked at them all together, that they have a shared pattern of early craniofacial development, and that they only diverge more radically later in fetal growth in particular. And moreover, when we looked at them more closely, we found that across all these different groups, that their shapes really converged around the time of facial prominence fusion, right? They're getting more similar at this time period. They're more similar to one another at that time point than any other time point. All right, this suggested to us that species that are as different as birds and humans might be more similar due to a common structural constraint. In other words, you think about the way in which you take individual facial prominences and you turn them into an integrated upper jaw, you really have limited solutions to how you approach that problem. And so that's what we hypothesized was actually going on with these guys. And when we went back to our developmental morphospace, we realized we could use it as a predictive tool as well, because the places in which animals don't occupy that morphospace actually predict something like clefts. And so that was suggestive that this was the primary structural mechanism that was going on there. And we experimentally confirmed that as well by manipulating embryos and then looking at how they occupy that new morphospace. So given the prevalence of cleft lip in humans as one of the most common structural birth defects, and the early time at which it develops, it seems that this kind of predictive model would actually be quite useful, right? So we sought to further develop the utility of this model by addressing three separate questions. One was we want to know whether these similar patterns of facial growth and constraint operate within species, not just between species. In other words, is it a statistical artifact or is it something that we actually see within individual groups? We also want to know whether these facial patterns were coordinated with underlying brain growth. We know the brain is a large player. In some ways, it's the landscape on which faces grow. So we want to know more about brain growth. And we want to then know if those things were true, can we use these developmental morphospaces to then predict and classify a range of normal and abnormal outcomes? So to answer these questions, we worked with our collaborators up in Calgary, longtime collaborators, and generated high resolution data in terms of using micro CT and iodine contrast allows us to get brains and faces. And we have a whole pipeline that allows us to do automated landmarking and pull out all sorts of cool data from these guys. We could build stage-specific atlases. These are not humans. These aren't birds. These are birds. Obviously, that's why there's a chicken there. And they're not blue. That's just color coding. We have blue chickens, and we could also extract their brains. We could build atlases for these things as well. So really, really nice data. And we could sample them in terms of our morphospaces, build morphospaces within species. And what we found in these nice rainbow-colored plots is that they had the same kind of characteristics that we saw within our comparative data set. They had the same kind of inverted shape, and it's true as well with the brain, which is not something that we had actually predicted or expected going into this. And what we found is that if we looked at them together, we looked at the brain and face together, that they exhibit the same kind of nonlinear pattern of growth, and that this is suggestive that embryos are converging in terms of their shape and their variability around the time of facial fusion, and that this is true of the brain as well, and that's coordinated in very strong ways. In fact, if you quantify the covariation between the brain and the face, you get the same pattern. You see that the brain is strongly predictive of the face and vice versa. And even more interestingly, and to go to the point of the actual title of this talk, when we look at the relationship between brain and face, what we find is that it seems to be a switch, a key switch that occurs at the time of facial fusion that's coordinated both between the brain and the face, which is describing allometric changes. Allometry is the study of relative shape, size between parts. And what we see is that there's a switch that's occurring there between what's going on in the brain and what's going on in terms of the face, and that this seems to be all coordinated around the time period of facial fusion. This is not just true of chickens. Early data suggests the same in mice. It's even true in humans. Okay? So what does this all mean? Well, it supports this idea that facial fusion is a critical developmental milestone, and that it's coordinated with the brain, and that these periods, these critical periods during and after are key areas, and during are key areas to investigate changes, say, in pathway activation. So you might, for example, predict that there are patterns of gene expression that are coordinated, that are switching at these key time periods as well. Okay. Ultimately, these data allow us then to test this model where we want to understand whether it's predictive or not. And we can test it in various ways. We might predict, for example, mutational variability will affect clefting, altering trajectories will affect clefting, as well as altering the relationship in the orchestration between brain and face. And this is all with the ultimate goal, obviously, to build better models for understanding cleft lip incidence and severity in humans. With that, I'd just like to thank Team Developmental Morphospace, Get Better, Zuzka, Feel Better, Diane, Wei, and Jay, and then, of course, longtime collaborators, Ralph, who's here in the audience, and Benedict, and, of course, the NIH and IDCR. Thank you.
Time for questions?
Sarah.
Yeah, I love the brain,
Yeah, me too.
but I also
It's
love
critical.
the peripheral nervous system.
Yeah.
And so with the cranial nerves, the innovation
Mhm.
of essentially the craniofacial complex, I assume during development of the chick as of mammals, what role are they going to have? Do you see any alterations in speed of innovation and all that sort of functionality, also brain function over time as well? So you're mixing that up with craniofacial, changes.
Yeah, well, it's a great place to explore. We have a ton of data on the brain. We really haven't even scratched the surface. Right now, we're just basically working with the structural aspects of it, but we could get down to the nitty gritty of actually visualizing where the nerves are and how they're interacting with those different parts of the face. I think one of the major insights from this is because we're often very face-centric, and we think about the face and facial prominences, how important they are. Just looking at the overwhelming size of the brain, how small the face really is at those time periods, and how much of a role the brain does play in terms of coordinating and orchestrating on structural levels, as well as signaling levels, and maybe even through communicating through nerve impulses, for example. So the orchestration is a key aspect. for sure.
That's all we have time for.
Thank you.
So
Thank
much.
you.
Okay, thank you. Our next speaker is Joseph Ordaz, and his talk is titled Digital Denture Workflow. Use that mic.
Okay.
Thanks.
No, as long as that was doing fine. It seems like doing fine. Okay, hello. Thank you all for joining today in person and remotely through Zoom. My name is Joseph Ordaz, and I'm a second-year prosthodontic resident here at the School of Dentistry. Today, I'll be discussing a digital denture workflow that I use to treat one of my patients. So before I get into the workflow, I wanted to discuss some advantages that milled digital dentures provide over conventional dentures, starting with less number of appointments needed to fabricate these dentures, and research has shown fewer number of post-delivery adjustments needed. These dentures also have superior strength since they are milled out of a pre-polymerized polymethyl methacrylate puck that is customized to each individual patient. These denture pucks are fabricated under extreme pressure, which makes them very dense and porosity free. Adaptation has been proven to be superior with milled dentures in a report published in the JPD by Dr. Brian Goodacre. In addition, in another paper published by Dr. Matthew Catadil in the JPD, he also found that digital dentures provide enhanced retention. Lastly is the advantage of reproducibility. If the patient were ever to need their dentures remade for whatever reason, the digital information can be accessed through the cloud. So I use the 3-appointment protocol to fabricate a set of digital dentures from Avident Digital Dental Solutions. Appointment 1 consists of taking an upper lip measurement, capturing digital impressions, and tooth selection. Appointment 2 is where a Wagner Tryon, also referred to as a WTI, is used to mold an accurate border extension, set anterior teeth, and capture the patient's VDO and centric relation. Appointment 3 was the delivery with the clinical remount to provide balanced occlusion. So at appointment 1, two lip measurements were made, one with the patient at rest and the other in full animation. For the intraoral scan, I use a vestibular retractor to reflect the patient's upper and lower lips to expose the depth of the vestibules. And since the mucosa is movable in this area, the retraction aided in stabilizing the tissue to facilitate stitching of the images taken by the intraoral scanner. Using a 3-shape intraoral scanner, I was able to capture a digital impression of the edentulous ridges. After this appointment, I uploaded the patient's STL digital scans, papillometer readings, and tooth mold selection onto the Avident website. So using the digital impression shown here on the left in blue, the Avident team was able to design a base for the Wagner Tryon that is shown here on the right in pink. So a base was also designed for the mandibular arch Tryon. So the Avident software uses the upper lip measurement to set the length of the anterior tooth. The software also relies on anatomical landmarks such as the incisive papilla. Central incisors on average are 8 to 10 millimeters anterior to the incisive papilla, and the canine tips are set in line with the center of the incisive papilla. Using the pterygoid maxillary notch and retromolar pad, the occlusal plane is established. So here's the design I received from Avident that incorporate all the landmarks and anatomical averages. So after my approval, these are the Wagner Tryons that I received. The upper teeth are set in a proprietary wax created by Dr. Steve Wagner that allows a clinician to easily make denture tooth adjustments by simply warming the wax in a water bath. I started with border molding the maxillary WTI with the heavy body PVS impression material. After that, I performed a wash impression using a light body PVS impression material. Patient's incisal edge was established using aesthetics and phonetics, and the patient approved the anterior setup. Patient's VDO was adjusted to provide adequate freeway space, and phonetics was also confirmed by having the patient speak aloud. After setting the VDO, I made an interocclusal record in centric relation using a rigid PVS material. The last step of this appointment was to capture a functional impression of the mandibular arch. The functional wash impression was made using a PVS material, and the patient was guided to close in centric relation at the predetermined VDO. So this is the finalized WTI. You can appreciate that the patient's border extensions have been captured, incisal edge position has been confirmed, and the VDO has been established in centric relation. After completing this appointment, I packaged the finalized WTI and shipped it to Avident to be digitally scanned. Image here is the final digital preview that has a lingualized occlusal scheme. The Avident online software allows for further customization if desired. Upon approval, the digital dentures are then nested to be milled in a custom denture puck. So this is the frontal view of the final denture that I received ready for delivery. Here is a lateral view of the final dentures. This is an intaglio view of the maxillary denture next to the maxillary impression for comparison. And this is an intaglio view of the mandibular denture next to the mandibular impression. So on the day of delivery, I checked border extensions, used pressure indicating paste to ensure there were no pressure spots. And you can see the patient's dentures had excellent adaptation. Using remount cast and a Facebook record, I was able to mount the patient's dentures on a semi-adjustable articulator. Once this was mounted, I was able to perform my clinical remount to obtain balanced occlusion. After this, I reviewed at-home denture care instructions with the patient. So here are the before and afters of the patient at rest from my frontal view. Here is a profile view of the patient at rest. You can see how the patient's facial form has been restored. Here is a frontal smile of the patient. Lastly, here's a smile from the profile view. In total, I had to see my patient for only one post-delivery adjustment. So for a workflow comparison, I just wanted to show that there's significantly more steps needed to make a conventional denture. And you can see how the digital workflow combines some of these steps. And there's also going to be fewer adjustments needed. On average, you need about 3 post-delivery adjustments with conventional dentures, and due to the well adaptation, studies have shown that no adjustments, and in my case, just 1 minor adjustment was needed. So I just want to say thank you to my program director, Dr. Sharma, for working with me on this case and designing a digital denture protocol to follow. I'd also like to thank Dr. Acharya for his support throughout the process, as well as I'd like to thank my patient and co-residents.
Any questions in the audience, please? Did you notice any difference between the patient's experience with this form of denture creation versus sort of conventional? Like, was there any sort of patient like personal like happiness with just the one visit, or did you notice anything?
Well, the patient, he's been wearing the dentures comfortably since, and he's really happy with the retention that he's getting and the fit. He says they feel great to him. Yeah.
Great.
But we obviously need to do more to see.
Yeah, great.
Okay. Okay.
All right.
Thank you.
So much. Okay, thank you. Our next talk is Christian Martinez talking about adaptability and value of a new approach to diversity, equity, and inclusion training for first-year dental students.
All right. Hi, everyone. My name is Christian Martinez, pronouns he, him, él. And my study focused on the acceptability and value of a new approach to diversity, equity, and inclusion, also known as DEI training for first-year dental students. So a little bit about myself. I'm actually bound to be the first dentist in my family. My family actually hails from a small village near Yoro, Honduras. And we struggle a lot growing up. I think my family and I were left out of oral health care because of the language discordance, as well as resource inequities. And so my experience has sensitized me to relate to those who have also experienced similar experiences as me. And so I think for me, it's important that we as future healthcare providers take a person's lived experience into account when we're providing care. All right, so an introduction, the ethnic and racial diversity of the dental workforce in the United States is not, it doesn't align with the general population of the United States. So that means that we as dentists need to be fully prepared to take care of patients with different backgrounds than our own. So we need to be culturally sensitive and culturally aware of the differences. So we also know that implicit bias may also affect a patient's outcomes, specifically in oral health. just because of the biases that dentists may have when making clinical decisions. So with all this being said, in the fall of 2020, UCSF implemented a new DI training for first-year dental students. And what that implied was that the training included 2 sessions during their first year. It included one during the orientation training in the fall, as well as one in the spring quarter. And so the training included things like introduction to oral health disparities. It included communication skills for equity, as well as breakout sessions led by facilitators and community building through personal narrative sharing, through the sharing of personal narratives. And so the way this worked is the first 2 cohorts completed the training. The second cohort completed last spring. So the class of 2024 was the first one, and then the class of 2025 completed it last spring. After each of those trainings, there was a survey sent out. And so there are 4 total surveys in total. And each of those surveys contained open-ended questions, as well as multiple choice questions using Likert scale. So people could put, I strongly agree, neutral, disagree, or strongly disagree based on the statement they were provided. And so what today's focus will be on the open-ended questions and analysis I did for those. And so as far as you can see here, the analysis that I did was first on the open-ended questions. And so the way that I did that is for each of the questions, I looked at all the responses. And from the responses, I looked at ones that shared similar context. So for example, if there were multiple students for a specific question that they appreciated the fact that people were being vulnerable, I put all of those responses together and coded them under a vulnerability code. After that, I went into the Deduce software and put in my document with the original codes that I started off with. And the Deduce software actually, it organized those codes for me, which then allowed me to get a more general and overarching theme that included multiple codes under one theme. So that's the way that was done. And then after that, after the analysis, the qualitative analysis of those open-ended questions, I then emailed 80 students from the class of 2024, which again was the first cohort to do it. I emailed them for an interview to see if they were willing to participate in an interview. And so I did that, and we ended up getting 8 open-ended qualitative interviews that were done, again, using the same methods with the Deduce and using that as well, and they were all done via Zoom. So here are the results from the response rates from the surveys. So these are all the response rates from the surveys. As you can see, 3 out of the 4 have response rates over 50%. Here's the qualitative, the first part of the qualitative analysis of the open-ended questions from the surveys. And so for that, we found 4 main themes for it. The first main theme was actually the appreciation of a smaller group where people felt like they had the chance to share freely. And so a quote from that was, talking to my peers about ways to oppose racism and discrimination. Also, we have learned that despite our differences, we have a lot in common as well. The second theme was the openness and vulnerability when people shared their narratives. I really valued the vulnerability and openness that my classmates share. I know it's hard to open up to people who you've barely met, so I'm honored to have been able to hear and be a part of people's personal stories. The third theme from the survey responses were having a safe space and being encouraged to share. So the facilitator guided our group with kindness, providing personal stories. She encouraged us to be non-judgmental and showed mutual respect to our team members. It was great. And the fourth theme from the survey responses were supporting and caring for people's experiences, as well as being open to continuously learning about other people's experiences, as well as learning about other cultures in general. So the quote for that is to never think that the problem is fixed, to always keep eyes and ears open to those who could use support and encouragement. So moving on to the qualitative analysis of interviews, again, it was with the class of 2024, 8 students. And we focus on the class of 2024 because they're now third-year students. So they're actually in clinic seeing people and treating patients. So we thought this was more applicable to them based on the interview questions we wanted to ask. So the first point that I want to make is for the interview section, a lot of the themes that we actually found from the interviews related back to the themes we found from the open-ended questions survey responses. So that was the first thing. And then the first main theme that we found that was different was that overall students do feel more comfortable with their class cohort because of the training. And as you can see here, and I wish it was higher than what I'm going to tell you, I would say like 20% of the time. I feel like people can be really vulnerable. It depends obviously in like context and setting and timing and everything. But I do feel like if we wouldn't have had that training, people wouldn't have opened up even that 20%. So I do feel like it's significant. And for that second main theme we found from the interviews was Students who feel comfortable treating patients with different backgrounds do so because of their past lived experiences. This could be work-related, personal lives, whether they moved a lot or just their culture and things like that. And then as far as the third main thing we found was that all 8 interviewees actually wanted more patient-centered DEI trainings. And sorry, going back to the second theme, I'll read the quote because I missed that. But I feel like most of my experience working with patients is because I've worked with patients before coming into school. But I know that some of my classmates don't necessarily because I hear it. So there's a need for more DEI trainings. Again, third theme, all of them wanted more patient-centered DEI training. And the quote for that is, as a third-year dental student, I would have hoped to have more cultural humility training by now. So in conclusion, students do feel that the trainings did help them connect with each other, but not all felt that the trainings helped them better be able to provide to patients with different backgrounds. There was also a need for more DEI trainings and long-term follow-up. A lot of them felt that it was just a one and done thing, and they never had any sort of similar DEI training ever again. Now they're seeing patients and they don't know what to do. And there was also a want and a need for faculty to go through the training themselves, just because there isn't a thing that's required for faculty and staff as there is for students currently. And then other than that, similar outcomes were observed from the School of Medicine's two-day DEI training. And then I just want to say that for me as a dental student and a child who regularly lacked access to regular oral care, I valued the DEI training, and I hope that the School of Dentistry will see the results and work to listen to the students and increases their support of long-term training in order to provide the dental school, in order to improve the dental school experience and our ability to treat the diverse patient pool we will be serving. And now I acknowledge all of these programs and the funds, as well as my 3 mentors, Dr. Davis, Dr. Hoff, and Dr. Taylor. None of this would have happened without your guidance and support, so thank you. I'll take any questions.
Hi there. I know you mentioned that the DEI trainings were implemented for class of 2024 and 2025. Were there any changes between that year and then the year after?
Yeah, good question. So there were a couple of minor changes throughout, but the biggest change that actually happened was during the first cohort for the class of 2024, the facilitators were actually from outside of school. And that was the same for the first session in the orientation for the class of 2025. But then while the class of 2025 was going through their first orientation, I believe there were faculty being trained that were related faculty of the dental school. So by the time we got to the spring quarter training for the class of 2025, there were faculty who were from the dental school that were facilitating that. And it's the same, that's going to be the same from now on. So I think that's the biggest change, the fact that now we have faculty from the dental school that are leading it, since it was important that the facilitator offered that space for people.
Thank you so much.
Yeah, thank you.
Okay, thank you. Our next talk is Tony Chu, who'll be talking about deconstructing calcineurin and fat signaling in species with distinct midfacial outgrowth.
All right. Thank you so much, Erica, for the introduction. Do you think chickens ever look at Leonardo DiCaprio and think, whoa, what a handsome dude. I wish I have his nose bridge, cheekbones, but instead I have this beak thing. So the question has to be asked, what makes us all look so different? So I started by focusing on craniofacial development in our favorite genetic model, the rodent. And what I'm showing you here is the developmental timeline in the mouse that requires incoming progenitor cells called neurocrests, which are highlighted here in red. And they have to migrate into the developing phase and give rise to these large structures called prominences. And here I've pointed out them, such as the frontal nasal prominences, the maxilla, and the mandible. And they must come together and fuse in order to properly form this developing phase. And for many of the images I'll show you today, I'll focus on this critical time window of embryonic day 10.5 and 11.5. One signaling pathway that is very important in embryonic development is calcium signaling, and calcium activates many downstream pathways. I'll be specifically focusing on one of them, which is the calcineurin NFAT pathway. So calcium first will activate calcineurin, which is a protein that removes phosphate groups from NFDC proteins. And this will promote NFTCs to go inside the nucleus. Once inside, the NFTCs will regulate expression of other genes. And there are 4 members in the NFTC family, including NFTC 1, 2, 3, and 4, kind of like my favorite K-pop group, Blackpink. So then to stop this NFTC's activities in the nucleus, other proteins like DYRK1A will add on this phosphate group, which will kick NFTCs back to the cytoplasm. So this balance of NFAT phosphorylation and dephosphorylation allows for this calcium dynamic dependent NFAT activity. So this DYRK1A protein is encoded by a gene in the Down syndrome critical region. And actually another gene that is also within this region encodes RCAM1, which is a calcineurin inhibitor. And working together, these 2 proteins lead to a reduction of nuclear NFAT, which lead to craniofacial abnormalities we see in Down syndrome patients. And it's unsurprisingly that actually loss of function of this pathway also lead to defects in craniofacial structures. So really understanding how this pathway is titrated is important here. Okay, so if NFTCs are Blackpink, then these other proteins are kind of like the producers and then the agents that work together to ensure the success of the team. So as a first step, I wanted to know, well, are these genes expressed at the right time and right place for craniofacial development? So I performed whole mount in situ hybridization of mouse embryos. And here, the red arrows are pointing to the face. You can see very strong purple signals in the developing phase of all of these different pathway members. And for the experts in the building, you can nicely see the expression with a frontal view highlighting the different prominences. Okay, then to do this in a more quantitative way, I also performed RNA-seq of mouse facial components, which is made up of 2 major cell types, mesenchymal and epithelial cells. You can see that DYRK1A, RCAM1, NFTC3, and C4 are expressed at high levels in both mesenchyme and epithelium, especially in the mesenchyme. And that suggests to me there's a potential involvement of this pathway in this early facial organization. So these observations prompt me to ask whether disruption of this pathway will lead to abnormal craniofacial development. In another word, if I take out some members from the Blackpink team, are they still going to be successful? So to test the functional involvement of this pathway, I first adapted a pharmacological strategy where I inhibited the activity of calcineurin using a drug called CSA. I used a series of paradigm. I'm only showing you the result of one here, where application of CSA during this critical time window clearly reduced the length of the head. And then I paired the pharmacology with a genetic strategy where I inactivated DYRK1A specifically in the mesenchyme. And once again, saw this dosage-dependent reduction in this head length. So these 2 strategies together show that proper titration of this pathway is really critical for craniofacial development. Okay, now you may ask, obviously this pathway is important in mammals, but what about other species that look completely different? You know, what about that handsome chick that was so envious of Leonardo DiCaprio? Well, it turns out chick have very different developmental trajectory than mammals. And more specifically, they have a elongated premaxilla, which you can see here in blue, and a reduced maxilla, which you can see here in red, which is the opposite of mammals and reptiles. So we began to interrogate the role of this pathway in chick. And once again, we turn to our old friend in situ hybridization. Okay, so you guys are familiar with the images below. I showed you guys earlier the C1 and C2 in the mouse. And it turns out we just don't really see them at all in the chick face. And the only one that's holding down the fort is C3, that is very strongly expressed in the face of the chick. And to our surprise, C4 is just completely absent from the chicken genome. So the question begs, what happens if we manipulate the expression of this pathway? Kind of like what I showed you with the mouse. It's as if, you know, in a parallel universe, Jenny is the only one left in Blackpink. So we ask, is she going to be successful going solo? So the 2 strategies I chose include gain and loss of function, either eliminating the expression of C3 or introducing C4, which is not there. And I couldn't help myself but to show you some very hot off the press preliminary data where I knocked down C3 expression in the chicken face. And it really appears that I'm getting a reduction in the facial length. Here I've highlighted the structures where I eliminated C3, which is the maxilla and the mandible. I still need to increase the number of animals I'm looking at, but it is extremely promising as the controls do not yield a difference between the two sides. So it does seem that C3 alone is able to direct facial outgrowth in chick. In conclusion, I showed you that the NFTCs are differentially expressed in chick and mouse, and by using genetic and pharmacology, disrupting this pathway lead to a decreased facial length in the mouse. And just now I showed you preliminary data in chick, also showing knockdown C3 alone could lead to a shortened face. So other than introducing C4 in the chick, I'm starting to look at a third species that has similar facial structures to the mammals, and that is the lizard. And I've already been able to see strong C4 expression there. So these are ongoing experiments. In the end, the project is really important for understanding how calcium functions during this normal and diseased facial development, including conditions such as Down syndrome. And maybe one day we'll understand what exactly makes a chicken look so different from Leonardo DiCaprio. Thank you. Oh, sorry, I forgot to thank my two labs, Li Chen, Georgia, collaborator from Schneider Lab, and my fantastic students. So thank you again.
Thank you, Bob.
I want to.
Thank you. Questions in the audience?
Great talk, Tony.
Thanks.
I was wondering if there's a clinical significance of understanding if this process is conserved across different species.
Yeah, that's a great question. I guess one thing if you think about this pathway is very clinically relevant in Down syndromes, right? So to understand how this pathway works, maybe can help us directly treat patients with Down syndrome in Down syndrome. As for comparing different species, I think kind of going back to what Nathan was talking about, understanding what exactly makes these different species look different and maybe leverage the different pathways that's being utilized could help us in treating our patients.
Tony.
Hi,
that was
Sarah.
Really funny and good.
Thank
So, you know, what is activating calcium signaling, right, during this time course? And so that's one question. You'd answer that.
Sure. That's a great question, right? Because I just showed you that calcium already inside the cell. So there are multiple pathways that can, multiple ways that can get calcium inside the cell, right? We can get it from the outside. So there's voltage-gated calcium channels that could lead these calcium to inside the cell. And actually, a mutation in one of the channels lead to Timothy syndrome, which also has a craniofacial defect. You can also get calcium from inside the cell. Right? There's intracellular storage, store-operated calcium entry that could let stores inside the cell to release calcium. So there are multiple ways of having calcium in the cell.
Quick question. Can you visualize calcium signaling in these cells?
Yeah, that's also a great question. So there's obviously calcium sensors that we could utilize to visualize calcium activity inside the cell. And that's definitely something I'm looking into and wanting to do in the future. Yeah.
Thank you so much.
Awesome.
Thank you. Okay, thank you, Tony. Our next speaker is Christy Huey. And her talk is titled Mitigating Bias and Application Review for Oral and Maxillofacial Surgery Residency.
Thank you. All right. Hi, everyone. Good afternoon. My name is Chrissy Huey. I'm a second-year oral maxillofacial surgery resident, and my mentor is Dr. Akshay Govind. He's the Highland Hospital oral maxillofacial surgery residency director. All right, so a little background, OMS remains among the most competitive specialties in dentistry, and there's no accepted process for fair application review as of now. So our objectives today are to describe common sources of bias in review for OMS residency and to propose tangible strategies to mitigate these biases. For methods, we conducted a comprehensive literature review, included papers from 1997 to 2021, and utilized UCSF and PubMed search tools. These are the following terms we used, and these are the topics we will be exploring today. So let's jump into demographics. So here we have a group of diverse names. Unfortunately, these authors found that names that sounded foreign, Black, and or female were evaluated more negatively than those that sounded white or anglicized and or male. And some authors also found that resume whitening had double callbacks for minority candidates, especially for Blacks and Asians. If I had gone by my Chinese name Xu Zuoxin, on my application, I would have been less likely to have been chosen for my position. And here we'll jump to photos. We'll pretend that we're a program director looking at these applicant photos. And what are some of the first things that come to mind? They all look like well-accomplished individuals. However, not all people may think the same way. And it turns out, females more than males are likely to have their IQ misperceived by evaluation of a photo. And equally qualified applicants with darker skin tone were given lower job suitability ratings than applicants with lighter skin tone. And in a study for dermatology residency candidates, those who had smiled, wore glasses, wore jackets and blazers were more likely to match, and female applicants with their hair down to their shoulders were associated with a positive match outcome. So we're looking at gender. Are women even going to dental specialties? And from this graph, we can definitively say yes, except for OMS, we're at 16.2%. But are women even going to surgery at all, you may ask? From this, we can tell from these surgical specialties, we see that representation in OMS ranks very poorly among all those in surgical specialties, and we're in between neurosurgery and orthopedic surgery. So various authors may have explored why this may be. Is it a bias in the app process? Is it lifestyle related? Is it a lack of mentorship and unsupportive environment? This last one hits home personally because I don't think I would be here today if it wasn't for my mentor, my oral surgeon, Dr. Tina Chang, back at the VA West Los Angeles during my GPR program. And we also have a slide on race and ethnicity. On the left, with the US Census from 2020, we're looking at the US population by race. And on the right, we're looking at the racial demographics of OMS residents who enrolled in 2018 to 2019. Black, Hispanic, and Native American groups roughly make up about a third of the US population, but only 9% are physicians. And as we can see on the right picture for OMS demographics, whites and Asians are clearly overrepresented, and we need to change this. Let's jump into application components. So as many have heard about the CBSE, this is a very difficult test. And prior to 2012, OMS programs only considered applicants with national board scores over 90. And when this went to pass-fail, OMS started using the CBSE exam, which is a shortened version of the Step 1 medical licensure exam. Higher CBSE scores are associated with higher odds of acceptance, although is only a modest indicator of Step 1 performance. It does not predict residency performance at all. In fact, the best predictor of OMS residency performance on USMLE is, you guessed it, dedicated time to study for the exam itself. And we'll jump into grades. Grades, class rank, and GPA are incredibly difficult to interpret as some schools use A's, B's, C's. Others use P's and H's or passes and honors. Some grade every course, only as others some. And applicants may be coming straight from school, from private practice, from GPRs or non-categorical internships, of which I have done both. Unfortunately, these two experiences are not well captured in their grades. And we'll jump to the CV. It is a challenge to differentiate between those with fewer high-quality projects and those with a greater number of superficial projects. Instead, scoring systems have been proposed looking for key areas of accomplishment depending on the mission of the institution. The quality of participation in projects listed on the CV should be explored further in the personal statement and in letters of rec written by mentors for these projects. Likewise, a scoring rubric would be implemented for the personal statement. And this is Dr. Govind's rubric, which he applies totally blinded to the rest of the application. So what we're looking for, who is the applicant? Why are they going to oral surgery? Why this program specifically? Their collaboration and excellence, their growth over time, the compassion for others, and organization of thoughts. But let's jump into letters of rec and gender. We'll compare these two candidates from the photo slide earlier. Male applicants are more likely to have increased word count, standout adjectives such as exceptional, reference to their awards, achievements, ability, and scholarship, whereas female identifying applicants are more likely to have shorter letters written for them, more likely to include negative comments such as doubt raisers, leave out their achievements and leadership potential, and more likely to have their physical appearance described, unfortunately, according to these authors below. For interviews, structured interviews will help to reduce biases, such as not having to look at standardized test scores and increase the diversity of the candidate pool. And some interview biases that CASA in 2021 had seen were applicants with accents, ethnic names, and overweight candidates. However, it's not yet known if there's a bias associated between in-person versus virtual interviewing, especially with the COVID pandemic the last couple of years. And let's next discuss the intersection of interviews and gender now. Women are 2.4 times more likely than men to experience gender-specific unprofessional conduct during OMS residency interviews. And women are more likely to receive inappropriate questions related to marital status, with the rates highest in surgical subspecialties. And there's overall bias against pregnant women in interview settings as well. So with all these biases and all the applicants and all the components of the application, what should we do? What can we do? So we borrowed a couple strategies from the authors that we found in these papers. And first of all, blinding the reviewers to CV and personal statement to numerical scores and photos. Also, increasing the number of women and underrepresented minorities in leadership positions. And also changing the CBSE and GPA from the initial screen to a more balanced part of the application review, as well as utilizing standardized letters of rec compared to narrative letters of rec in order to reduce gender biases. Using scoring systems for the CV, letters of rec, and personal statements, as well as using structured interviews to fairly compare one applicant to another. In terms of limitations, we did use broad literature sources, which may not all apply to OMS. Explanatory models for trends are difficult to deduce. And if all these measures are considered problematic, then what can we use to compare applicants with? For future studies, we plan on blinding reviewers in OMS to certain parts of applications and asking them to score other parts to review biases held in our field. And in conclusion, a more thorough understanding of these common sources of biases during the app review will help program directors and selection committees mitigate the impact on the field of OMS. I just wanted to give a quick shout out to my second year class. Always appreciative that we can always share our different cultures and experiences because of how diverse we are. And shout out to our female majority as well. This is what we're aiming for at a national level. And these are my references.
Okay, thank you. Any questions from the audience?
Hello, first of all, I really appreciated your talk. Thank you. And I'm also someone who's had many of those experiences you described. So it was nice to see that visibility in the setting. I wanted to ask if you had any thoughts about what applicants can do if they've had a discriminatory experience or had one of these negative experiences happen to them during an interview. Is there any way you can think of to have a follow-up or some type of mitigation of that experience afterwards that can help add equity if we're not able to catch all those issues with the interviewers? I hope that made sense.
Yeah, no, that did. No, thank you for that question. That's a really good question. I think if anything, we should hold our interviewers and interviewees accountable. If anything, we should have a survey email go out after every interview just to make sure that everyone's experiences were equal. I think that's definitely a good thing to incorporate to the process.
Wondering, do you think it's worthwhile to include a DEI implicit bias training prior to allowing interviewers to interview applicants?
Yeah, no, that's also a good question. They did a study where people or interviewers who had training before actually were less likely to discuss discriminatory topics or any coercive topics. So I think DEI training is very good for all programs.
Thank you so much.
Yeah, thank you.
Thank you. Okay, thank you so much. And the last talk of the session is Riva Gestre, who will be speaking on TikTok and Dentistry, a content analysis.
Hi, everyone. Good afternoon. My name is Riva, and this summer I worked with Dr. Jean Calvo to look at the fastest growing social network, TikTok, to analyze the content and quality of dental videos. So oral health is the most unmet healthcare need in adolescence. It's an impressionable time to create long-term habits, so dentistry needs innovation for oral health promotion for this group. Emerging social media platforms such as TikTok can provide opportunities for health communication and patient education. TikTok currently has, as of today, the hashtag dentist has 23.9 billion views total, and 63% of American adolescents use it on a daily basis. And so while TikTok does contain a lot of videos about dentistry, the quality of information is unknown. So I performed an observational mixed methods descriptive analysis from June 28th to August 19th, looking at the top 15 daily most liked videos with the hashtag dentist every day, as well as the top 100 all-time most liked videos with the hashtag dentist on a bi-weekly basis. I accessed TikTok through a private browser, used Qualtrics to enter my data, and did a data analysis. And so the data collected included uploader and video demographics, as well as content and quality analysis. So for uploader demographics, I looked specifically at the uploader engagement, such as the number of followers or the total likes an uploader had. And I also split the two into two groups, uploader type, dental professional versus non-dental professional. And this was determined by whether in their name, their username, or even their bio, if they had doctor, DDS, DMD, or a description about their career. For video demographics, I looked at video length, views, likes, comments, and shares, specific treatment discipline, if applicable, as well as if props were used. For content analysis, I looked at the intended validity, the narrative reliability, video production, video sentiment, and video content of each video. For quality analysis, I used validated consumer and patient education methods such as PEMAT, which stands for the Patient Education Materials Assessment Tool. And this helps to determine whether patients can understand and act on information. There are example questions up there, and they're answered on agree, disagree, or not applicable. The higher the score, the more understandable and actionable material. So if the video made its purpose completely evident, if it used common language, used clear illustrations, and chunked information into short sections. And then if a video depicted treatment, then I moved on to use the Discern instrument, which is a questionnaire used to assess the quality and reliability of information on treatment choices. And this was a continuous rating scale from 1 to 5, with 1 being no, 3 being partially, and 5 being yes. So the first section looks at whether the publication is reliable, and the second section looks at whether, looking at the quality of information presented. So on to the results. The results that I had were split into dental professional and non-dental professional. So first, here are 2 examples of videos that I analyzed on TikTok. Think to yourself, what would be the content and quality of these videos? I don't know if the music will play. This says, me after destroying your gums with my sharp instruments and telling you that your gums bleed because you don't floss. And then the second video.
For me, I really like corn.
What do you like about corn?
It's corn! A good corn Vietnam. It has the juice. It has a more beautiful thing. It's corn! I can tell you all about it. I mean, look at.
So in that video, you can see they're offering examples of oral health supplies you can use for the days that you're just too depressed to take care of your own teeth.
For me,
Sorry.
I really like corn.
All right, technical difficulties. Okay, so in my inclusion criteria, I was able to include around 1,000 videos. There were about 500 that were excluded because they were either not in English or not relevant to dentistry. Looking at user demographics, there was no statistically significant difference between the number of followers and total likes between dental and non-dental professionals, but interestingly enough, there was a significant difference in terms of whether they were verified. So dental professionals were less likely to be verified than non-dental professionals. And non-dental professionals were those that could have been like patients, parents, comedians, meme pages, or just social media influencers. For video demographics, there was a significant difference in terms of the average number of views and shares between dental professionals and non-dental professionals, and that dental professionals received more views and shares compared to non-dental professionals. But there is no difference between the average number of likes and comments. Looking at content analysis, the intended validity looks at whether the video is informative or not informative. A not informative video would be one where it's a dentist, they're just dancing or singing, but they're not providing any education. So 75% of videos uploaded by dental professionals were informative, compared to 58% of videos uploaded by non-dental professionals were informative. Narrative reliability looks at whether the uploader intends to inform the viewer with correct or incorrect information. So 71% of videos uploaded by dental professionals intended to inform with correct information, while 7% were incorrect or misleading. For non-dental professionals, 28% intended to inform, while 23% were incorrect and misleading. And again, the ones that were listed as not applicable were videos where they're just dancing or singing, no educational value to them. This was actually the most surprising statistic I found. So the most common video sentiment in videos uploaded by dental professionals was actually fear at 40%, followed by neutral and humor/parody, which both came in at 25%, and only 10% of videos elicited empathy. Whereas for non-dental professionals, the most common video sentiment was humor at 56%, followed by fear, neutral, and empathy coming again at last. So PEMAT, again, as I said, looks at the understandability and actionability of the material. There was no significant difference between the PEMAT scores, the median PEMAT scores. They both had a median score of around 70%. So the data here and the data I'm going to show in the next slides, I use the median instead of the mean because the data was it was not normally distributed. So the median was a better representation compared to the mean. So at 70%, if that's a grade, that's like a C. So they get a C, both groups overall in terms of understandability and actionability. And then if videos depicted treatment, I use the discern instrument. So in terms of reliability, the maximum score possible was 35. And even though there was a significant difference between dental and non-dental professionals, 13 out of 35 is around 30%. So that's really not a reliable score. And the second discern section looked at the quality of information on treatment choices. So again, maximum score was 40, but the medians were 11 and 9. And even though there was a significant difference, 11 out of 40 is again around 30%. So these are the videos I showed earlier. These were both posted by dentists. And so you can see the video on the left, the sentiment was fear, and it was also incorrect or misleading, trying to tell viewers that the reason that you bleed at the dentist is because I pick and prod at you. Compared to the video on the right, the PEMAT score was actually 100% and the sentiment was empathy. The video on the left of the PEMAT score was 62% in terms of understandability. Not again. Okay, so in conclusion, dental professionals were less likely to be verified, but they on average had more views and shares. But unfortunately, the most common video sentiment was fear. Both groups were presented understandable and actionable material, but in terms of reliability and the quality, it was very low. So videos with the hashtag dentist do not have reliable nor high quality information, no matter who is posting it. And dental professionals should be taking advantage of TikTok's widespread use to engage with the population, especially adolescents, to distribute and improve patients' access to oral health education and understanding of treatment choices. I'd like to thank the Lee Heisen Fund and Dr. Jean Calvo for mentorship and guidance. Thank you. Question in the front.
Hi, Reva. Thank you so much for your presentation. You did a great job. I guess I have a general question since you looked through a lot of TikTok videos. What's kind of like your general kind of comments on dentists who are posting TikToks that aren't informative at all? Like for instance, dance challenges.
I think it depends on like what the content is in the video, because there were a lot of videos where they're just dancing or singing, but sometimes they're like completely harmless. Like they're not trying to present misleading information. And if anything, they show the general public that, hey, we can have fun. We can participate in TikTok challenges. But when it gets to the point where they're trying to mislead, like with the video on the left, that's when it can be harmful.
Thank you.